Hey guys, happy snow day. Sorry I didn't hop on here sooner, but I am going to share my screen with you, go through the presentation and show you the notes. So, okay, we are now sharing my screen. So you guys, all you have to do is go to slide 23 of this presentation. I do have the notes to match, and for some reason it, oh, hold on one second. And then let me show you the notes. That's my homework. Just disregard that. Here we go. Here are the notes. All right, so here are the notes. If you're able to print them out, great. If you can't, that's okay. So just do this at your own discretion. If you want to write it word for word, you can, or we're just going to have to be creative, and I apologize. We're going to have to figure it out if you don't have a printer at home. So let's get started with the presentation. All right, so we're going to go into mitosis and the cell cycle. We can skip the essential questions because your notes are going to start on this page. So this is a little review, and what I like to do is compound each chapter and unit. You guys will notice there's a tiny bit of review as we overlap the different chapters. So according to the cell theory, all living things are made up of one or more cells. The cell is the basic unit of structure and organization that performs life functions, and all cells come from pre-existing cells through cell division. So we've learned that you can't create cells like from molecules in a lab. So these are your 3 cell theories. And again, I'm just going to zoom through these slides. You guys can pause it for as long as you need to take notes. All right, so cell division. This is kind of bothering me right here. Why is it like that? Whoops. Anyways, okay. Cell division. Oh no, what happened? This is funny. Okay, here we go. Cell division is the process by which a partner cell divides and gives rise to 2 or more daughter cells. Mitosis, division of somatic body cells to produce 2 genetically identical daughter cells. So you guys, we're going to learn the difference right now between mitosis and meiosis. Meiosis is the division of sex cells, sperm or egg, to produce 4 genetically unique daughter cells. So when I use the word somatic cells, those are like your body cells. So if someone broke their arm or got a cut, your body would regenerate those cells through mitosis. The lesson focuses on mitosis, or cell division of somatic body cells of eukaryotic organisms. Why do cells divide? Cell division is necessary for formation of new cells for the growth and development of organisms, replacement of damaged cells, like I mentioned, replacement of old and worn out cells. So you guys will notice your skin is the largest organ of your body, and it sloughs dead cells, constantly replacing those cells, as an example. So new somatic cells are created to facilitate growth and development. When you are injured, new cells are produced to replace the damaged cells, and new cells are produced to replace the worn out and dying cells in your body. The cell nucleus. Yep, I wanted to move that. Okay, the cell nucleus. The nucleus stores and protects the genetic information of a cell. The nucleolus, the spherical structure in the nucleus that produces and assembles the ribosome. So remember, the nucleolus is where the 2 subunits of the ribosome are created. So some of this is a little review. The nuclear envelope is the double membrane structure that surrounds the nucleus, and the nuclear pore, the pores in the nuclear envelope that control the passages of the substances between the nucleus and the cytoplasm. Genetic material. All right, so now we're getting into the good stuff of mitosis. So genetic material, DNA, contains genetic information that an organism needs to develop, function, and reproduce. So I noticed a couple people just put nucleic acids, that chromosomes were made of nucleic acids. So it's important to put that it's deoxyribonucleic acids. Those are two different things. Okay, so chromatin, uncoiled stringy complex of DNA and protein. Chromosome is the dense and compact bundles of chromatin that make it possible to separate DNA precisely during cell division. Sister chromatids, two identical halves of a chromosome that have been replicated in preparation for cell division. Centromere, the region of a chromosome where two sister chromatids are joined at the center. So if you look at the center of your chromosome, that is your centromere. Moving and organizing genetic materials. Centrioles and spindle fibers are two cell organelles that assist with organizing and moving the chromosomes and chromatids during cell division. So here's your centrioles are here, the little yellow guys, and all these little dark lines are the spindle fibers. Centrioles, a pair of structures that organize the spindle fibers during cell division in animal cells. Plants do not have centrioles. Spindle fibers, microtubules that help to separate and move duplicated chromosomes during cell division. Okay, the cell cycle. The cell cycle is the series of events that occurs during the formation of two genetically identical daughter cells, the somatic cells, from the division of a mother cell. During cell division, cells perform these processes in an organized and predictable series of steps. The cell cycle is broken up into three parts. So here's interphase in the green, okay? So three parts. So these are steps within interphase. G1, S, and G2 are steps within interphase. If you hear the question, what is the longest cycle seen in a cell? It is interphase. Then there is an interphase prepares you for cell division, and it also does the activities of the cell, the normal activities of the cell. Then there's mitosis here in orange. So that's cell division, and cytokinesis is the actual separation of the cytoplasm to complete cell division. So here's interphase, the longest phase of the division, and prepares the cell for mitosis. The cell grows, DNA and organelles are replicated. The cell is not dividing. Cell engages in normal cell activities. Interphase is broken down into three stages. G1, a cell grows in size and duplicates organelles. S, synthesis, cell synthesizes a copy of DNA. When the chromosomes are replicated, and G2, cell grows larger and reorganizes its genetic material in preparation for mitosis. So G1, S, and G2 here. Okay, so interphase, the prefix inter means in between. This will help you to remember that interphase occurs in between the times the cell is actively dividing. So as you guys can see, you have your chromatin here. The little dark spot here is the centrioles, and then your nuclear envelope and nucleolus. A cell spends about 90% of its life cycle in interphase. Nucleus and nucleolus are well defined. DNA is loosely packed into long chromatin fibers. Cell grows and replicates DNA and organelles, and the cell performs normal cell activities. Next, mitosis, or the M phase. Mitosis is the stage of the cell cycle where the cell is organizing duplicated chromosomes and is actively dividing into two identical daughter cells. So I like to think of mitosis like this. So mitosis is broken up into four phases. When I was in college, I would memorize it by PMAT, or if you want to memorize it with interphase and cytokinesis, I can't even say it. We would say IPMATK, and then that is the first letter of each phase, and that was our acronym, and that's how we memorized it for tests, because we weren't allowed to have notes during a test. So mitosis is prophase, metaphase, anaphase, telophase. So if I ask you what that is, you can think of the acronym PMAT to help you to remember mitosis. The goal of mitosis is to produce two genetically identical daughter cells. Okay, prophase, the prefix pro means before or first. So prophase was always first. This will help you to remember that prophase is the first stage. Chromatin condenses and chromosomes become visible. Centrioles start to move to the opposite poles of the cell. Spindle fibers extend from the centrosome, the area where centrioles are located. Nucleolus disappears and the nuclear envelope breaks down. Prophase. Early prophase, chromosomes condense, mitotic spindle begins to form, nucleolus disappears, and the nuclear envelope dissolves. So this is early prophase. Late prophase, sometimes called prometaphase, spindle fibers attach to centromeres, creating a kinetochore. Kinetochore connects two centrioles, chromosomes begin moving. Metaphase, you can think of metaphase as middle. The letter M is the term for metaphase. You remember the chromosomes line up in the middle during metaphase. Chromosomes line up along the center of the cell on the metaphase plate. So this is called the plate, metaphase plate. Spindle fibers coordinate the movement of chromosomes. So here is a real animal cell with metaphase going on. And you guys may have, the root tip. You may or may not have remembered seeing this in the root tip that I showed on the large microscope. Chromosomes line up on the metaphase plate. It's typically the shortest phase of mitosis. Anaphase, the letter A in the term anaphase will help you to remember that duplicated chromosomes are moving away from each other. During anaphase, chromosomes separate at the centromere and move away from each other. Chromosomes move along spindle fibers to opposite ends of the cell. Once anaphase begins, each sister chromatid, so these are the sister chromatids, is considered an individual chromosome. Each side of the cell contains a complete set of chromosomes. So once they're separated into a sister chromatid, this is one set of chromosomes when it was the attached chromosome, one set of DNA. Yeah, and an individual chromosome. When they were attached, they were considered duplicates. Anaphase, sister chromatids are separated at the centromere. Chromosomes move to opposite ends or poles of the cell in anaphase. Telophase, condensed chromosomes unwind and spread out into a cluster of chromatin. Nuclear envelope reforms. The nucleolus becomes visible. Spindle fibers break apart. The prefix telo in telophase means end or completion. This will help you to remember that telophase is the last stage of mitosis. Telophase, a nuclear membrane forms around each group of chromosomes, so you can start seeing it here. The cell has two identical nuclei. Chromosomes unwind in chromatin. Mitosis ends after telophase, and the next phase of cell division is cytokinesis. So cytokinesis completes the process of cell division by splitting the cytoplasm and dividing one cell into two identical cells. Each daughter cell contains an identical set of duplicate chromosomes. Cytokinesis occurs differently in plant and animal cells. You'll probably notice why we have a cell wall here. Cytokinesis in animal cells, the animal cell membrane draws inward, creating a cleavage furrow. So this is called the cleavage furrow. Cytoplasm is pinched in two parts. Two identical daughter cells are created. Cytokinesis in plant cells. A rigid cell wall surrounds the membrane of the plants. A cell plate forms between the divided cells and gradually develops into two cell membranes that separate the daughter cells. So you can see the cell plate here. The new cell wall then forms between those membranes. All right, so you guys can stop here. You do not have to go to comprehension check because we are going to be doing a lab that does your comprehension check. Oh, this can be your last slide, which I believe is slide 23. And I hope you enjoy your snow day.